Okay, so 5 minutes to go. Yeah, I would like to quickly introduce you to the Metagenomics-Toolkit. It's mainly developed by Peter Bellmann and Benedikt Osterholz, shown here in the middle. So what is the Metagenomics-Toolkit? It is a powerful and scalable workflow for whole genome shotgun-based metagenomics. And first of all, it's a full end-to-end MAC pipeline. So it does all the steps from raw Illumina and ONT reads up to binned and annotated MACs. It provides some additional features like the recovery of unassembled MACs, plasmid detection, metabolic modeling, and also dereplication in co-recurrence. And lastly, it's optimized for cloud environments because cloud resources are expensive and especially high memory virtual machines are even more expensive. So those resources should be used as efficiently as possible. So yeah, let's start with the basic workflow. So this is a workflow that gets executed for each sample which you provide. The workflow itself is structured in modular components, of which maybe like the input module and the quality control module is one. Most of these modules can also be run independently if you need to run them independently. And yeah, the rest is just a standard workflow for MAC-based metagenomics from quality trimming, assembly, binning, classification, and annotation using best practice tools, I would say. Yeah, one feature which is unique for the Metagenomics-Toolkit is the plasmid module, or it's two modules actually, an assembly module and an examination module, which provides functionality for plasmid assembly, the detection of plasmids, and the comparison of detected plasmids to known plasmids in databases. Another important feature is the ability to recover unassembled genomes in a microbial community. So due to low coverage, you might have genomes that are not in your assembly. Then you can provide reference genomes as an additional input, map unmapped reads from the binning to those references, and then they are processed in all the downstream analysis of the pipeline. Another feature is the computation of genome scale metabolic models. They are computed for each MAC. The reconstruction of these metabolic pathways is done by the tools CARFME and GAPseq, and after that, you can identify microbial interactions by using Smetana in the toolkit. And yeah, besides this per sample workflow, the toolkit provides an aggregation workflow, and this aggregation workflow combines the results from all the single sample workflows together. And the first step in this aggregation workflow is the dereplication, where we just cluster all the bins from all the assemblies together. We only consider medium quality MACs per default. You can adjust that to your likings. Then we do a clustering based on ANI values, which you can also adjust. And after that, we choose a representative for each cluster, which we base on completeness and contamination values. And these representatives are then the common ground for the abundance analysis, where we map all the reads from our samples back to the representative, and then we have comparable abundance values. Based on these abundance values, we compute a co-recurrence, either as a covariance estimation with Big Easy or a correlation coefficient with Spearman. Okay, now I'd like to introduce a short example project where we analyze the members of the sewage core microbiome. This is a publicly available data set from the global sewage surveillance. It consists of more than 750 data sets with around 10 terabase pairs of data. So quite a large amount of data. And the bottleneck in the pipeline is often the assembly, because it requires a large amount of RAM. So we implemented a peak RAM prediction, where we apply a machine learning approach to predict the peak run prediction of an assembler. And by this, we can reduce the requirement of high memory hardware because it's expensive, and also improve the parallelization of all the assembly compute jobs. For the parallelization itself, we use a Slurm cluster because we are processing large amounts of data set. We scale out to a cloud environment. And there we have many machines available, also different sized machines. And for the setup of the Slurm cluster, we use the BibiGrid framework, which starts a cluster in the cloud and is able to auto-scale. So it shuts down large machines when they are no longer needed after the assembly, for example, and starts new machines when they are necessary. Okay, just to give you an idea how much resources were needed for this project. This were approximately 200 VMs with 28 cores and 210 GB of RAM for around 60 days. Of course, there were larger machines involved for larger assembly tasks and smaller machines for simple computational tasks. So the overall, this resulted in more than 17,000 MUX, and after dereplication, around 3,500 were left. Then we estimated the abundance by mapping all the reads to the replicated MUX, and could find that there are in some areas of the world there are some species which are that define the core microbiome, whereas in other parts they are not present. Like the red one, for example, is only present in Europe in almost all countries. So the size of the circle means how many countries contain that organism. Okay, so the last feature I want to mention is that all the data that is produced from the pipeline is exported as a JSON file. The JSON file can be imported into the exploratory metagenome browser. And there you can inspect each sample. You can filter for certain taxa which you are interested in. You can inspect pathways, have a closer look on your assembled MUX. And if you want, you can even go down to a contig level and have a look on all the genes of a specific contig. Okay, and lastly, I would point you to our GitHub repository where the toolkit is available for download. We also have a link to guides and tutorials that get you hopefully easily started. The workflow is also integrated in the cloud-based workflow manager Clown, which is developed in Bielefeld. And there you can really easily start the workflow. You can upload all your data in an S3 bucket, and with a few mouse clicks, start the workflow, get the results in an S3 bucket, share it with your colleagues, and you're done. Okay, yeah, then thanks for your attention, and maybe there's time for a question.
Hi, I'm not very familiar with this field, but the previous presentations were showing that multi-sample binning is better than single-sample binning. So I'm wondering if your tool can be extended to take multiple sample input.
Multiple sample binning? Yeah, probably. So we currently it performs all the tasks for a single sample, but Benedict already implemented co-assembly, and I think in this part it might be implemented.
Actually, the answer is yes, it can.
Yeah.
These kind of core pipelines are getting very popular. I know them. So do you have any plans to put your tool into this core pipeline? People can use it more easily?
No, currently not. So we're planning to keep it as a separate pipeline contribute because of the different modules that go beyond the scope of the MARC pipeline. I think the MARC pipeline is meant to be assemble MARCs, annotate them, and the additional tools we have are not in the scope of the MAC pipeline, as far as I'm concerned.
Yeah, I think it's not 100% compatible with NF4 at the moment.
Yeah, that's of course.
So we have to see if we can make it NF4 compatible. Okay, 2 more questions and then we can stop.
Mine's quick, and I guess you probably know the answer already. How easy would it be to incorporate viral hunting mapping into your
Viral hunting mapping. So
viral hunting mapping? So you can make mags out of, you can make viral genomes from metagenomes.
yeah. What would you need to do to so as long as if there's a tool that does it?
Yeah, there's tools to do it. There's a bunch of different tools.
Yeah, if there are tools that do it, you could just put it in the right place in the pipeline, and so a single tool is quite easily integrated. And also, if the database is not limited for I don't know, for and costs money, then it can also be, because all the databases we use, we have them in our S3, and they are downloaded automatically. And if the database is not freely available, then it gets a bit more complicated.
That's a question about databases.
Yeah.
Hi, good, great work. And I have a question. Is I think one feature of your work is you said there is plasmid assembly, right? You can do plasmid assembly in your workflow.
Yeah, it does. It uses what is it? SCAPP is a tool. So it uses it re-evaluates the assembly graph of the assembly and tries to find some loops in the
Actually,
assembly.
my question is you develop a specific assembly by yourself, or you use existing algorithm. So you use SCAPP to assembly, and you mentioned that you align it to non-plasmid to identify if the plasmid is correct, right, correct or not, right?
Yeah, firstly we run these detection tools, and so for the comparison we use a PL.
By my colleague.
Okay.
I also do plasmid assembly. So my question is, if you align them to non-plasmid, then are you able to identify novel plasmid that is not available in non?
Yeah, we aligned to as we use this PLSDB.
You align the plasmid to non-reference database.
Yes.
Then are you able to identify novel plasmid that is not available in the non?
Yeah, if it doesn't map to the database, then we have.
Okay. I think there's a lot of novel plasmid that is interesting. Maybe it can be interesting for exploit in the future.
Yeah. They are identified if they don't map, then they you get that information. Yeah.
Okay, thank you very much to all the speakers of the session. And we meet